How do we compute genotypic and phenotypic proportions in crosses where individuals have aberrant ploidy? And let's start by computing the proportions of different gametic genotypes in meiosis in a trisomic. And we have a genotype, a trisomic of genotype big A over little a. And so we have three chromosomes here, a big A chromosome in green, a little a chromosome in orange, and a second little a chromosome in blue. Now recall that when you have meiosis in trisomic, two of the non-sister chromatids, let's say the green and orange ones, will segregate to one pole, while the third one will segregate to the second pole. And this will result in one gamete having two chromosomes and the other gamete having only one chromosome. But this is not the only way the gametes can be produced. It's also possible that instead of the blue segregating alone, you could have the green segregating alone or the orange segregating alone, and the remaining two would segregate to the other side. When the green segregates alone, then the blue and the orange are on the other side, whereas when the orange segregates alone, the blue and the green are on the other side. Now we can go ahead and label the alleles. The greens are big A's, the orange are little a's, and the blues are also little a's, giving us the following genotypes. So here's a diploid gamete written as big A over little a. Here is a haploid gamete, so it has a genotype of little a. Then you have little a, big A, little a, and little a. Now, since the univalent segregates randomly, it chooses which side in a random fashion, all of these possibilities are equally likely, and therefore they're all 1/6. And we can say the probability of having a haploid gamete of genotype little a is 1/6 plus 1/6 or 1/3, and the probability of having a haploid big A gamete is just 1/6. The probability of having a diploid big A little a gamete is 1/6 plus 1/6 or 1/3, and finally the probability of having a little a diploid gamete is 1/6. Another way to compute these proportions, the gametic proportions, is using a branching tree diagram. So a gamete could either be haploid or diploid, and the probability of either situation is a half, then haploid gametes could be big A, or they could be the orange little a or the blue little a. All of these occur with a 1/3 probability, since they're equally likely to segregate alone. Now, if you are a diploid gamete, you also have 3 possibilities. You could be big A with the orange little a, or you could be big A with the blue little a, or you could be little a over little a, and all of these also have a 1/3 probability. And applying the product rule, we see that each of these gametes have a 1/6 probability, and then we can apply the sum rule to compute the probability of big A little a diploid gametes or little a haploid gametes. Let's practice with an example problem. In Drosophila, trisomics and monosomics for the tiny chromosome 4 are viable, but nullisomics, which means there's no chromosome 4, and tetrasomics are not. The B locus is on this chromosome. Deduce the phenotypic proportions in the progeny of the cross between, this is a cross between a trisomic B plus B plus B with an individual homozygous for the mutant allele. The significance of the statement that trisomics and monosomics are viable, but nullisomics and tetrasomics are not, is that when we are looking at the progeny, if they are diploid or trisomic or monosomic, we should include them in our phenotypic proportions. But if we see any nullisomics or tetrasomics, we should exclude them from the analysis because they're not viable. So we have to compute the different proportions, different types of gametes that this trisomic individual is going to produce. And we can do so by drawing a branching diagram. So the gamete could be haploid or diploid with probability of a half, and then it could be if it's haploid, it could be B plus, or it could have the other B plus, or it could have the B, all with a probability of a third. And if it's diploid, it could be B plus over B, or B plus over B with the second B plus, or it could be B plus over B plus, all with a probability of a third. And that gives us, with the product rule, a probability of 1/6 for each of these types of gametes. And we can now use the sum rule to say the probability of a haploid B plus gamete is 1/6 plus 1/6, which is 1/3. The probability of a haploid B gamete is 1/6. the probability of a diploid B plus over B gamete is 1/6 plus 1/6 is 1/3, and the probability of a diploid B plus B plus gamete is 1/6. We don't need the branching diagram anymore, so we can get rid of it, and we have our proportions of gametic genotypes over here. So the other individual is homozygous for the little b allele, therefore the progeny will have 4 different genotypes. We will have the B plus gamete from the trisomic fertilizing a little b. We will have little b. These two will be diploid progeny because they are the result of fertilization of 2 haploid gametes. But then we will also get trisomic individuals, B plus B over B, and finally B plus over little b. And since all the gametes of this individual are little b, the probabilities are simply the probabilities of the different gametes from the trisomic individual. And so this is 1/3, this is 1/6, this is 1/3, and this is 1/6. We need to determine the phenotypic proportions, so we should assign phenotypes to them, and the only one which will be mutant is the second one. Everything else is wild type because it has at least one wild type B plus allele, and usually in such problems we assume that the mutant is recessive, and so 1/6 will be mutant, which means we will have a 5/6 will be wild type, and so we will have a 5:1 wild type is to mutant ratio. Suppose that in guinea pigs, ABO blood types are determined as in humans, where one IA allele, so it will be IA over IA or IA over little I, gives blood type A. IB over IB or IB over little I gives blood type B. IA and IB are codominant, so if you have both IA and IB, then the blood type is AB, and little I over little I is the blood type O. During a set of routine crosses, you notice something unusual. Repeated matings between a certain female with blood type AB and a certain male with blood type A produces F1 guinea pigs of all 4 blood types in this ratio: 5A, 2B, 4AB, and 1O. And our task is to determine what are the most likely genotype of the parents. Well, so the female is of blood type AB, while the male is of blood type A. Since the male has blood type A, they must have at least one IA allele, whereas the female, being blood type AB, must have both an IA and IB allele. However, the progeny have blood type O. That means this progeny are little I, and therefore both the parents must have a little I allele. That makes the female at least a trisomic and the male diploid. Let's see if this combination will give us the desired proportions. So we have to determine the genotypic proportions of the gametes of the female and the male. The male is easy. It's diploid, so it's going to be half IA and half little I. For the female, however, being a trisomic, let's make a branching diagram, and you could get a gamete that's either haploid with the probability half or diploid with the probability half, and this haploid gamete could be IA or IB or little I, and each one of these has a 1/3 chance. Now, the diploid also has 3 genotypes possible if you could have IB over little I. That is, if IA segregated to the other side, or you could have IA over little I. That is, if IB segregated to the other side, or you could have IA over IB if the little I segregated to the other side, and all these three also have a 1/3 chance of occurring. Now we can actually just add the genotypes of the male onto this branching diagram and compute the proportions of the progeny. So this IA could fertilize, this haploid IA gamete could fertilize either the IA or the little I gamete from the male, and these would have a probability of half. The same goes for the IB and the little I as well. The IBI, little I, could also fertilize either IA or little I, IA or little I, IA or little I. And all of these branches have, if we use the product rule, are half times a third times a half. And so they're all 1/12 probabilities by the product rule. And we can now go ahead and write down all the blood types. For instance, if you are IA, you are blood type A. Or if you're IA, you're blood type A. Or if you're IA, you're blood type A. If you are IA, you're blood type A. Or if you're IA, you're blood type A. Therefore, we have 1, 2, 3, 4, 5, a probability of 5/12 to be blood type A. Next, we could go and consider blood type B, which you could do in two ways. You could be IBI, that's blood type B, or you could be IBII, that's blood type B, and therefore we have 2/12 progeny who are blood type B. Then we can go and look for blood type AB, which you would have IBIA, that's blood type AB. Then you have IBIIA, that's blood type AB. And all of these are going to be blood type AB because they have both an A allele and a B allele, and that means you will have 4/12 that are blood type AB. And finally, that leaves us with just one situation, which is over here, little I, which will give us blood type O, and that will be 1/12 blood type O. Since the genotypes we inferred give us the observed ratios, these are the correct genotypes of the parents. Let's compute the genotypic proportions of the gametes from a tetraploid. So we have 4 chromosomes here. And recall that during meiosis, there are 3 ways that these chromosomes can pair up. You could have either 2 bivalents, in which case you get 2 diploid gametes, which look something like this. You can get a quadrivalent, which also results in 2 diploid gametes, like the one shown here. And the third way that these 4 chromosomes can pair up is as 1 trivalent and 1 univalent. In that situation, since the univalent and the unpaired, the third chromosome in the trivalent segregate randomly, you could end up with 2 diploid gametes, but you can also end up with aneuploid gametes, where you either have a triploid gamete or a haploid gamete. Now, since these aneuploid gametes are not going to produce viable progeny, we can essentially ignore them from our analysis, and we just need to consider, when we are looking at the genotypic proportions of these gametes, how many ways are there to produce diploid gametes from these 4 chromosomes. And so the question we are asking is, how many ways are there to choose 2 chromosomes out of 4 chromosomes? And there are several different ways of doing this, but one thing that's easier is to give them these chromosomes numbers. And we can just start to write all the different ways in which you can pick 2. Choose 2 out of 4. How many ways are there? Well, I could pick 1 and 2. I could pick 1 and 3. I could pick 1 and 4. Then I could pick 2 and 3. 2 and 1 is the same thing as 1 and 2 because the order in which you pick them doesn't matter. All that matters is that you have the first chromosome and the second chromosome. Similarly, after 2, 3, I could pick 2 and 4, and then there is a last possibility, which is to pick 3 and 4. And you can see here that there are 6 ways, and all the 6 ways are equally likely. So they have all a probability of 1/6. And we can draw this out as well. So for instance, here I have 1 and 2 and 3 and 4 as a combination. So I have used up this and this. Next, I could consider having 1, 3. So I have 1 and I have 3. And by necessity, on the other side, I will get 2 and 4. So I have 2 and 4 over here, and that leaves me with 2 other possibilities, 1 and 4. So 1 and 4 and 2 and 3. And all of these possibilities are equally likely. So there's 1/6, 1/6, 1/6, 1/6, and 1/6. Now, let's say that this tetraploid had a genotype big A, little a, little a, little a. And that means I could say this chromosome is a big A and everything else is a little a. Little a, and little a. And therefore, I can go and write down the genotypes of these diploid gametes. For example, my 1, 2 gamete is going to be big A, little a. The 3, 4 gamete is going to be little a, little a. The 1, 3 gamete is big A, little a. And the 2, 4 gamete is little a, little a. The 1, 4 gamete is big A, little a. And the 2, 3 gamete is little a, little a. And what this shows us is that there are only 2 diploid gametes possible over here. One is big A, little a, and that has a probability of 1/6 plus 1/6 plus 1/6, which is equal to a half. And the little a gamete has is diploid and has a probability of 1/6, 1/6 plus 1/6 plus 1/6, which is also equal to a half. Next, let's try and do a problem. A tetraploid plant of genotype big A, little a is selfed. And so the cross we're doing is big A, little a crossed to big A, little a. And the question asks, what proportion of the progeny have the recessive phenotype? Now first, let's work out the genotypic proportions of these gametes, and the problem is simplified because both the parents have the same genotype, and so we need to just do one calculation. And so one way of doing this calculation is to assign these chromosomes numbers 1, 2, 3, and 4, and then determine the probabilities of choosing each pair from the 4 chromosomes. For example, the probability of choosing 1, 2 is 1/6. The probability of choosing 1, 3 is 1/6. The probability of choosing 1, 4 is 1/6. The probability of choosing 2, 3 is 1/6. The probability of choosing 2, 4 is 1/6. And finally, the probability of choosing 3, 4 is 1/6. Having written this down, now we can go and fill in what the genotypes are going to be for these gametic genotypes. So for example, 1 and 2 is big A. 1 and 3 is big A, little a. 1 and 4 is big A, little a. 2 and 3 is big A, little a. 2 and 4 is big A, little a. And 3 and 4 is little a, little a. Now we can use the sum rule to determine the probability of each gametic genotype. So we can say the probability of big A is just 1/6. We can say that the probability of big A, little a is 1/6 plus 1/6 plus 1/6 plus 1/6, so 4/6 or 2/3. And finally, the probability of little a gamete is 1/6. Having done this computation, we don't need these numbers anymore, and now we are ready to compute the proportion of the progeny who have the recessive phenotype. Now, in order to have the recessive phenotype, you have to be have all little a alleles, all recessive alleles, since the general assumption in such problems is that even one dominant allele will give you the dominant phenotype. Therefore, we need a little a gamete from the first parent, and so we're using the product rule here. We need a little a gamete from the second parent, and since the probability of a little a is 1/6, the probability of getting a little a gamete from the first parent and the second parent is 1/6 times 1/6, which is 1/36. And so 1/36 of the progeny are going to be mutant, while 35/36 will be wild type. And therefore we will get a 35:1 ratio of wild type is to mutant.